I'll take a couple of minutes to introduce Dr. Sasi Kotagiri. Dr. Shashi Kumar Kotagiri completed his doctoral study at the Center for Cellular and Molecular Biology in Hyderabad, one of the premier research organizations in frontier areas of modern biology in India. So at the CCMB, he worked on chronic myeloid leukemia and skeletal muscle differentiation. He was also a recipient of the Indian Council of Medical Research, or the ICMR Fellowship. Now, after obtaining his PhD degree, he received the Department of Science and Technology, or the DST, National Postdoctoral Fellowship to work as a postdoctoral fellow in the Indian Institute of Chemical Technology, IICT, on developing therapeutics for diabetes. Currently, he's working as a postdoctoral fellow at the prestigious MD Anderson Cancer Center in Houston, Texas. He is exploring non-small cell lung cancer pathogenesis to discover therapeutics. So it gives me great pleasure to hand over the screen to Dr. Shashi Kotagiri. Sir.
Hello, can you hear me?
Yes, sir. Yeah. You can start sharing the screen, please.
Yeah. Sure. Hello? Hello?
Yeah, sir, it is visible.
Yeah. So, good evening, all of you. Actually, this is the first time I'm giving a talk on a summer school organized by Bhaktivedanta Institute. I thank for giving me the opportunity for the lecture on this origin of life. Actually, my research is not on origin of life, but origin of life is basically the studying the molecular biology and other molecules, so biochemistry involved. Because of that background, and then now currently, as you know, I'm working on cancer, but this is an interesting topic which fascinated me from many years, and it's like a good opportunity. It's my privilege to speak for the summer school in remembrance of Sujipto Ghosh, Dr. Sujipto Ghosh. So before, it can be, you can, I present my talk, so you can stop if you have any doubts, because so it will be clear if you clarify the doubt, and then you can proceed further. So is it okay? Can I proceed now? Yeah. So how did life begin? So this life is a very interesting topic of many researchers, include physicists, biologists, and chemists. Many physicists like Schrödinger, Wigner, and so many people have, even though not of biology related, life is related to biology and all. So many, Werner Harber, and chemists like Watson, Crick, Miller, so many people have got attracted to this, to know what is life, because we generally live our whole life and we don't know the definition of the life. Then it's like a, yeah, it's a joke kind of thing. So uh, so what are like what we don't know, we have advanced technologically and scientifically in this 20th century. So there's still almost many questions, important questions, fundamental questions that were unanswered, that was published in Science, like with the title 125 unanswered questions, what we don't know. So among that, many questions, like people sometimes, whenever they meet an Nobel laureate, they asked, okay, how to get a Nobel Prize? So which question to be addressed? So for younger generation or whoever interested to get a Nobel Prize, they can solve any of these questions. I think some will be solved. So if they solve, I think it's so. Um, so regarding that, important questions, two important questions are very like fascinating questions. Like it attracted many people from the ages, from philosophers, scientists, and poets. So uh, the two important questions is what is life, how and where did life on Earth started. So what is life is not the topic of this today's presentation. How and where did life begin? So just like um, did life started? How did life begin? So I said this is a topic of research of interest for ages. So initially, people thought of one theory called abiogenesis. They have observation. Later, it was named as abiogenesis theory. Like life, people, scientists, they have observed a natural phenomena occurring in the nature. So they have observed that, and mice at the garbage and frogs from the mud. So frogs generally they live it, and mice they go for the garbage to get something. So seeing this observation, they said that the living things are origin like are coming from the non-living things, like mice from the garbage or frogs from mud. So this is an earlier concept. They thought that way, but a cell theory, opposite this abiogenesis theory, cell tells that cells comes from pre-existing cells. So this theory existed for some time, and then later came the biogenesis theory, which have given a blow to the abiogenesis theory, mentioning that life comes from life, living things. Yeah. So in this happened in 1861, conducted by famous Louis Pasteur experiment. So it have completely changed the thinking of other people. So Louis Pasteur conducted his swan neck experiments, where this is a simple experiment. He taken the broth so that these have the nutrients for the organism to grow, and then he did is like he um, heated the broth so that whatever the already organisms there in the broth will be killed. So next he had uh, he had um, did heated this broth into this Van Neck flask. One of the this one is the this and the other one is this. So what he did, he broken one of the swan neck of this container. Then he observed that there is no life here for how long he stored, but here because this is completely closed. So no organism can come from air into this broth to grow. But here, as swan neck was broken, so the flies, whatever organisms enter inside the broth, they feed and life comes from that. So he proposed that life comes from life, but uh, uh, not from non-living things. You can see here he observed that no organism evaluated from here. So this biogenesis theory have occupied the concept for many years, and so other concept at the time had by Operon and Halliday is that so how this life began. Okay, Louis Pasteur said uh, that life come from life, but how the initial life came before the earth, like after when the earth formed, and how the it led to the life. So, but it should start with some, at least some simple cell like bacteria or something. Then we can think of the whole cell. So, the initial thought of was that there is an electric spark, big electric spark, which gives the power for the molecules to combine. The initial molecules, as you know, the predominant biomolecules of H2O, that is water, hydrogen, ammonia, and methane. So because of this lightning and all, there is like chemical reactions occur between these, and biomolecules are produced, which apparently fall into the like with rain into the soup called primordial soup. They hypothesize that okay, this is the place because this is nutrient rich and it have all the some nutrients. So life originated from this primordial soup. But to do experiments for this, they have carried different experiments, but one success they got. But here, what we have initially and what is our aim. So, thing is that initially molecules were there, like precursors. So, after UV light or something, they should form a monomer, like amino acids, nucleotides, and carbohydrates, which are the basic uh, units for the polymerization to happen, which the polymers are the requirements for the life. So these amino acids, nucleotides, and carbohydrates, they polymerize into protein, oligonucleotides, and polymerases, which ultimately, when they combine together with lipids, because lipids are important, they form proto cells. These were the concept. So scientists, as I said, scientists did some experiments, and one is a path-breaking experiment is Stanley Miller experiment. So what he did is he taken water, methane, ammonium, and hydrogen, and as I said, like they used it to mimic the prebiotic condition. They use the electric discharge, and he like they heated the water for circulation so that water will move into this wherever the spark happened, and it will. So they used to analyze, collect the samples here, and so they send some the gas inlet here and collect. So they have analyzed that uh, amino acids are formed, uh, basic amino acids are formed. That is, two amino acids are formed by this experiment. So then it's a revolutionary experiment because at the time people have except some senses people have given this no one have given this proof. So these people thought, okay, this is like how living things coming from molecules that like maybe delicate. This is the it might have happened that because we have got this precursors, then we can get the polymers and further. But this is the initial um, uh, like setup which Miller used during the 1950s to and created building blocks. Is like a is a path-breaking finding, and then so this is in 1950s. So after that, many people tried to synthesize because I mean basic amino acids is not the life. We should get so many other things like ribonucleotides and lipids. So all together will form a life. So after almost 60 years, then the scientist called Sutherland. He is from MRC laboratory. What he did is further showed ahead that one with using the hydrogen cyanide, he used the hydrogen sulfide, and what he made is like copper and all, and he synthesized some of the precursor molecules like lipids, amino acids, and nucleotides. So this is what Miller also did, but that is not just amino acid, but here Sutherland have generated all these precursors with this common chemistry. So he proposed that with this common chemistry, life have originated. Like might any chance that this is the reason. So we can further have polymers and get that. So what the experiment he did, finally what he got, ribonucleotides, amino acids, and lipids. So these are this he got from the from precursor molecule of hydrogen cyanide and its derivatives. So he used the UV light and this copper for the photo redox cycling, and he got this precursor molecules. But this also not to the extent of what we say they're like monomers. Our aim is polymers and polymers to protocells. That's what how the origin of life happened. If that is the theory they have to prove. So that means from molecules it should form a single cell, and single cell to it will form embryo, because single cell formation is very difficult. Then after that it should give rise to embryo. But how complex is the cell? Many of you know might be knowing, but I just give an inner life of cell, how complex, how integrated it is, and yeah, you can. Oh, sorry. I think yeah. Sorry, just but I showing the video.
Yeah, sir. Got hang, I guess.
Hello.
Yeah, is it uploaded in the slides or?
Yeah. It is just I have to click that. I have to minimize this.
Okay, you can stop the.
Shall I stop and the one?
Yeah.
Okay. Just.
While sharing, select optimize for video. Share screen, then share computer audio, and also select optimize for video.
Okay.
Just after clicking the share, in the pop-up that appear on left bottom corner, you can see those two check boxes.
Okay, optimize. Okay, video clip. Yeah. Can you see?
Yes. We are seeing the presentation.
It's a long video, but I'll try to drag it. Just
we're able to hear.
You can hear the sound.
Yes.
Yeah, this is the complex of the cell. Like
If possible, make it full
hello?
screen. Yeah, if possible, make it full screen.
It's not full screen?
No, it is not in presentation mode.
Hello?
No, it is not in presentation mode.
Oh, okay, sorry.
Yeah.
Now? Shravan, is it?
Yeah, maybe some of the other team members can confirm. I'm not able to.
No, yeah, I'm also not able to see.
Yeah.
Actually,
What are you editing?
Where is video playing? In the PowerPoint itself or in another application?
Yeah, I attached it there. I copied there in PowerPoint.
Where are you seeing? Are you seeing in our screen on your screen?
I am seeing. Okay. I'm seeing on my laptop.
In your laptop. While sharing, did you select the whole screen or have you selected only the PowerPoint? Yeah. Okay.
You need to go to presentation mode to play.
Either you can share the link, we can play.
Okay.
That would be better. You can try to play the video separately and then come back to the slides. That is more convenient.
Okay. Yeah, sure. Okay. I think.
Yeah, we can proceed.
Long video also. Yes. It's a long video. I think better and time constraint also. I will just stop that and.
Yes, please. We can go ahead with the slides.
Yeah. Yeah. So I don't know, like I couldn't do it. So it's very complex integrated system where many suborganelles like mitochondria, um, this lysosomes and nucleus and so many endoplasm, so many things are there and microtubules. They will be working in a like a busy factory. So many intricate things will be happening on. So how this beautiful cell with a highly integrated system can occur with a simple molecules. So further addressing the theories were proposed on origin of life. So some of the theories which are occupied the prominent place is the deep sea hydrothermal vent theory, panspermic theory, and RNA world theory. So here the deep sea vental hydrothermal theory, what does it state? Like it says that okay, life have originated um, in this deep sea hydrothermal vents where there is a conditions of alkalinity and so there will be on so like per ferrous sulfide, some compounds will be there, minerals, which can help for the uh, life to form in the conditions of alkaline and altered pH. So this is one hypothesis, and as you know, that Nick Lane will be presenting. Dr. Nick Lane will be presenting tomorrow. He had some good important observations. So this is one place in New Zealand. So it's a hot spring. So at this, they have found that there is RNA-like molecules, but they couldn't find any life here. So this what briefly the deep sea hydro vent theory proposes. And another is the panspermic theory, which is also called cosmic theory, which states that life originated from in different places, and it might have come with some asteroids or comets. This asteroid hit the Earth, so they bring some life or molecules when they uh, come. So this is one theory, and this come under all astrobiology. And I think morning you heard from Vikram, Dr. Vikram Singh. So this also showing some signs of life, but it's not yet proved completely. So, and now apart in the astrobiology only, they are thinking that life, because Mars and Earth have similar earlier conditions of environment. They think that life might be there in Mars also. Just they want to send. So US sent a rover called Perseverance. So you can see here this is a Carter. It's like a, it's called Zero Carter. So where you can see here there is a, this is like previously this was, these are some river kind of thing where life can be there. So this is a magnified image which you can see here, um, like some liquid have flown and then it's frozen like that. So they have sent this robo, so it will collect the samples. Recently they have sent in 2021 after the COVID, they send it. So what the thing is, they bring some samples from the this Carter and to check that. But for returning, this rover will collect the Perseverance, will collect the samples, but for getting back, it will take 10 years because there is no funding much to be for this research and all. So coming to the important theory, the RNA world theory, which uh, scientists have proposed some of the proofs to say that um, this RNA is a precursor molecule which have given to the life. So what is RNA? RNA is a ribonucleic acid. So it have specific functions which are not there with either protein and DNA altogether. So as you all know, DNA is a genetic material. So for the life to continue, it should have the information kept with it. So what to be done with for the future with this information. So generally, people thought initially DNA is the genetic material that precursor molecule for the life to origin. But DNA alone can't do anything. It need a help of protein to unfold the helix, and then single strand RNA will be formed from DNA when it unwind. So because for that protein is required. But for the protein synthesis to occur, all mRNA should be found, and so all these uh, enzymes, proteins required. So initially, people thought, okay, DNA is the um, uh, material precursor which given to life, but without protein, nothing. So which came first, the protein or DNA? So they're looking for a different molecule which you have the performance like have the genetic, which it can store the genetic information and act as a template for protein synthesis, and also function as an enzyme. So without enzymes, no biological process will happen. So giving this, because of these functions of RNA, people thought, okay, RNA might be the precursor molecule first formed on the earth, and then it led to the formation of the life. So this is central dogma of life. This happen in every cell. Without that, uh, no function is performed. So as I said, DNA is double helix structure. This is very stable one. By when it when RNA polymerase unwinds this, the transcription, this phenomenon is called transcription, where the single strand RNA will be formed. Because it's single strand, it's not much stable than as DNA. But Using the reverse transcriptase, this RNA can be made into the cDNA reverse process, and from this RNA, with the phenomenon of translation, when with the help of ribosomes, proteins are formed. Proteins are like basic amino acids in a chain polymer. Then they fold and form the protein. This fold will help for them to function as an enzymes or antibodies or receptor signaling. So our body is made up of mainly proteins. But for the protein to form, it happens in the transcription translation step. So here is the RNA intermediary between this DNA and protein. So people said that, and scientists said that ancestral RNA world might have used this RNA, which can form as enzymes, which I show in the next slides. So the hypothesis, but that RNA is a precursor molecule which may give the life because it have the genetic information and that ability for give rise to the protein with the other when translation happens. But for this we need a proof. But Altman and Chet discovered that RNA can act as catalyst. This is an extra function they have discovered. For that, they have given Nobel Prize in chemistry. Then whole world like Francis Crick and Watson. So many people, it attracted many people's thought process, and then they said, okay, this RNA world might be the precursor at the prebiotic condition, which may further led to the life because it have the genetic information and the enzyme like property catalysis. Because like things to happen, this catalysis minimize the reaction, hastens the speed of the reaction to happen. So and here as a proof, scientists have identified that R18 polymerase. This is the structure of as I previously showed, I showed a single strand of RNA. It generally it won't be there like that in the nature because there is no complementary thing. So it form bonds among itself and form a like ribosomes. It may be functional when they fold it. You can see there are different folds here. It may be functional or non-functional, but some things have the property of functionality so that it can act as an enzyme. So this RNA polymerase, there's initially on the earth, people propose that, okay, there is cooperative between these RNA polymerases. This RNA polymerase is nothing but ribosomes acting as a polymerase with the different structure of the like a folded structure. So they say different RNA polymerases are there. There are interaction between them. Each one catalyze the other one and then change, modify so that different molecules arise by that. So this is called autocatalysis. Generally, if a substrate A will be there, for it to become substrate B, we will add a different substrate called C. Then A will give to the B in presence of C. But here there will be only A and other things B. So these are all RNA molecules or C or D. So these RNA molecules, they perform the same autocatalysis, help each other, but they won't come a protein or some other thing to make these changes. So they worked in cooperative, and that's why there's like a the chance that life came from this RNA is a hypothesis. So, but how this is the like I just showed here in a just different number of RNA ribosomes. But here this is the structure of the ribosomes. Agaricus recombinase. So you can see there are different strands of RNA folded here and forming complementary pair with other strands. So these different strands come together, perform a single function of catalysis, either to synthesize RNA like that. So what is the current research going on ribosomes? So in I think 2002, there is a ligase called R3C ligase ribozyme. Ligase is like it will ligate two strands. Suppose to form a polymer, there should monomers will be there. So two monomers should be ligated. So ligation should be will happen with the enzymes. So this RNA form as ribozyme, which have the function of the ligation. So it was said that okay, this ribozyme acting as enzyme activity, they are forming the monomers into the like polymer chain. So later, people have recently shown that this RNA, this ribosome act as RNA polymerase and cause the amplification of RNA, so that this way they say that predominantly the RNA might be there in the prebiotic condition. But here they have synthesized nucleotides. Uh, this was published in PNS by George. So he says, but the length of the nucleotide are very less, 20 nucleotide. With 20 nucleotide, 25 nucleotide, it's like hard to form like a functional ribosome. So, and another important observation they have is the ribozymes catalyze the RNA synthesis using a triplet building blocks. So they say, okay, which led to the formation of a polymer. And finally, one thing, the this RNA polymerase is making the uh, its copies of its own. This is a recent finding in 2020. So it makes in, suppose A is there, A is a ribozyme. So this RNA ribozyme will make A itself. So, but there is, because in host system there are like enzyme proteins are there to proofread any misadded nucleotide is there in the DNA sequence. So, but here it is because of this mutations, there is no earlier precursor enzyme in the earlier word to proofread this one. There are mistakes, and after many copies generated from the copying of itself are very less efficient in performing its function. So they have advanced this shown the advancement in science, but they have some drawbacks for this theory. So what have they have shown here is like till now research over the years that active ribozymes or ligases were formed from the random sequence of the single-stranded RNA, but this not prove that like it led to the polymerase and so many other carbohydrates should evolve by these reactions, or and then lipids should require, then only the life will form. But till now there is no enough proof for this RNA hypothesis theory. What they have shown is the with this ribozyme having the property of catalysis. That is the important they have mentioned. So NASA showed in mentioned in 1996 that there are significant difficulties in this for this concept to prove that life originated from RNA, because chemical fragility of the RNA. That is, RNA is, as I said earlier, like it's a single strand, and for that, even when we are working in the lab, we have to use specific gloves, because even with the hand, we have this enzymes for cleaving the RNA. So we should be very careful for that. But they have to, this RNA is so sensitive and single stranded, and even for temperature storage also, we store them at -8 and -20. So, but at the precondition, prebiotic condition, we don't know like how it is and harsh it is because of the UV and so many things. So they have proposed that okay, this might be not the chance. uh, RNA could be the precursor molecule for the life, and it's also narrow range of catalysis activity it have. As I said, there are many single-stranded RNAs. They fold on them, like fold internally. Many, very few will get the catalytic function. So to start the life, and there are so many other questions. Scientists are facing a problem regarding that. So think one some of the questions I have: how specific are these first formed ribozymes? Because in enzyme activity should be any specific. The protein, they show specific reaction, because if A is given as a substrate, it should form as B. So if there is no specificity for the ribozyme and the substrate, it can't give the B. So that is the one of the problem they face. And how life sparked like came by a single polymerase, or I have mentioned that autocatalytic set of ribozymes. How they said okay, this is happening, but how is that happening is not proven. And okay, these ribozymes are formed, okay, and if this is encapsulated in a lipid droplets, so lipids, as you all know, that cell is made up of lipid outer covering. I think Dr. Taur have shown previously in lipid membrane and all. So this, when RNA is capsulated in this lipid bilayer, is it how, because they say RNA is there, but there should be lipid to be encapsulated, and other nutrients, other polymers should be there in that vesicle, so that life can be started. But um, there is no idea about it. And for the body to function, we need energy source as ATP, and whatever or other and mostly we use the ATP energy. But for this monomers to form as polymer, we require this energy. So where from the energy is coming? What is the source? And how is it uh, possible to link this energy source for the metabolism synthesis of RNA? So that's also not answered. So, and then it should be thermically driven, this synthesis of RNA RNA molecules, or it replication. Some more things are that, like first replicating ribosomes in the absence of, because these are like I said, from DNA RNA will be coming. So this DNA acts as a template, and RNA is produced. But without having a template, initial DNA is not there, only RNA is there. How it's initially the RNA formed? So next is that this these ribozymes, they act as polymers, and they act for template for the production of the next RNAs. But it can't simultaneously happen at the same time. So, and one important is the substrate availability. In the prehistoric biotic condition, uh, okay, what is there? Lightning is there, and some precursor is there. Okay, RNA formed, but RNA formed somewhere else, and so many RNA have fallen on Earth because of they suppose they form a polymers, and uh, but for them the substrate availability, for things to a reaction to happen, there should be in some compartmentalized structure, so that the availability is there. So they change. It lead to the further downstream process of polymerization and all. So, but there's no any confinement or compartmentalization to these reactions to happen. So, and this prebiotic environmental factor, they might be harsh for this RNA to survive because of this less stability. So these all the scientists facing the problem to say that RNA world theory is the ultimate, like is the main theory for this origin of life. So sorry.
Dr. Sasi, we have about 5 minutes more for your speech before the Q&A. There's a
How much time?
5 more minutes before your Q&A.
Yeah. Okay. Yeah. So the goal is the protocells. From there, where the cells form, but started with the molecules. We like till now research have shown that there is formation of amino acid or nucleotide amino acid from the Stanley Miller one and nucleotides um, uh, from nucleotides and ribonucleotides and some amino acids and lipids from the Sutherland experiment from MRC laboratory, but it not led to the formation of the polymers. Polymers are, so polymers are the next stage for the to start the life process. So till now research have gained like showed that we are at the stage of monomers, and some polymers are, but they are limited of length. So is it possible to synthesize life? Okay, you take the replicase RNA and then vesicles. Vesicles are made up of the lipids. They form when you keep a lipid in water. They're not miscible. So with a special technique, they form these vesicles. So you keep this RNA replicase all together in the compartmentalized vesicles. They form RNA encapsulated vesicles. But initially, if this form, then what about the um, uh, other basic polymers require like mm, carbohydrates and proteins, so they all should be there, then only the life will be formed. Just combining the RNAs or whatever the vesicle is there at the prebiotic condition, life can't originate. So uh, like this mystery of a minimum cell. So scientists have tried from precursor like RNA world, whatever it have not given a cell till now, but Craig Venter Institute constructed a cell with a synthetic genome. That means they used the Mycoplasma mycoides, and they have in 2010 they have synthesized this constructed the cell, bacterial cell. It's because it's a simple cell. If you can't create this, it will be difficult for constructing a eukaryote cell, because eukaryote cell is so complex. So they have what they have done is they have reduced the genome size, and then in 2016 they have made a synthetic 3.0 cell. So they said what they have shown is the minimum number of genes required for its cell for the survival and replicating. But here, this is not the creation of life from the precursor molecules. What they have done is they have deleted using bioinformatic analysis. They have deleted some of the genes um, uh, and then check which are essential for the survival and all. So they have synthesized the oligonucleotide synthesis for gene synthesis, and the genome they constructed, and then they cloned in the yeast for to make the more copies of this genome, and then this is the genome they kept in a already living cell. So this already living cell is having this genome with many genes, and they have kept this genome inside, and they have removed this already existing genome uh, uh, of the bacteria. So this they say okay, we have synthesized the life constructed. That's why it's called constructed the life. So whatever already is there, because it's like how simple example, home is there, someone left the house and we have taken that rent for it, but the house is already there, so just we have entered there. So it's not say that like it's not that, because this, even the bacterium have a outer what is it, cell wall. It's very complex to make. So if this not came from the precursor molecule, just they have just taken the genome and kept the different genome. That's why it synthesis. And what till now the cell not not originated from any precursor molecule. And some scientists say, pioneer scientists suggesting that life is beyond molecules. So one of important a quote from Werner Arbor, a Nobel laureate in medicine and physiology. He made an important discovery, like ribone restriction enzymes, which revolutionized the science. Each and every person will be using that enzyme. So that important work is his, and he thought that the primitive cell requires hundred of different biological micromolecules like lipids, proteins, and all. So how this complex structure of the cell um, can be formed by these molecules together is like a mystery. So he thinks that there is a possibility of existence God is there to answer this problem. And a famous pioneer in the origin of life, Orgel, he proposed that there are many tenable theories, many speculations about this origin of organic material from the primitive earth, but there is no compelling evidence for this life originated from the uh, non-living things, precursor molecules of like RNA or DNA. So the famous quote, like Crick, Francis Crick, Nobel Prize winner, he says that he like writing a paper on the origin of life, it will make him fascinated, and he will speculate so much because it might happen that way, because no one has seen the prebiotic condition. But whatever the little experimental knowledge, they speculate. And so it's like facts are less and speculation is more. So for him, writing a paper will be very difficult. So because paper writing is based upon the facts, if many speculations, the reader also can't have the fun in reading that. So um, so he confessed that I don't want to write it, but that's an important topic of research. It will attracted from him to write more. So conclusion is that many theories of origin of life have been proposed, but none of the theory is fully accepted. Here, accepted means like they have given promising, some of the promising were like might be there, like because they see, for example, of RNA, it have the property of self-replicating and passing the information for the further generation with the formation of the new RNA molecules and all. But we are at the stage of just uh, some monomers and some chain of polymers. But life is much beyond that. It's very complex, forming a simple cell. So, and they just till now just making the precursor, but not much achieved by that. And many scientists claim that life is beyond molecules, uh, and one require an alternative paradigm of life is required to propose to say that how life originated on Earth. Yeah, thank you. And this is a complex city of a cell having the nucleus, mitochondria, and this endoplasmic reticulum, Golgi, and some centrioles. Just um, and this in cytoplasm, and this is just a three-dimensional art. But as I said, I have a big video of the inner life of cell. But I think even I have it occupies like 8 minutes or something. Yeah, this is the complexity of the cell which have to be achieved. Yeah, thank you. So, and if you have
Thank you, Dr. Sashi.
yeah,
Can we have some questions? We have
yeah,
got,
yeah,
we can take a couple of questions. Can we unshare Dr. Sashi's slide so that we can take a look at the hands that are raised? There is a question by Nikit. Nikit, can you please unmute yourself? Nikit?
Yes, now everybody can hear me, I think. Thank you, sir, for your lecture on this origin of life. As you said that so many philosophers are so many theories, like there is something related to cancer cell also. Cancer cell, nobody knows how it originates in body and then
hello,
fights the
Nikhil.
body
Your
mindset
audio is not
We can't
clear.
hear you very well. Nikit, can you
percolate.
adjust your audio, please? It's not very clear. It's breaking.
Yes.
Okay.
Can you hear me?
Yes.
Yeah.
Yes.
Say your question.
Actually, the question is related to like how we can relate origin of life with cancerous cells. The origin of cell is also unclear, like first RNA or DNA, then RNA. It was proposed since it is more accepted as a catalyst. Then like cancerous cells also percolate in the body, and cancer cell also nobody know how it originates and then starts fighting with other organs of the body also, and it creates its own variations and different kinds of tumors also within the same body it creates. So how can we relate cancer cell to the origin of life and its variations then? Means any research regarding cancer cells also and this kind of thing model in this.
Yeah. One thing is that these two things are completely different. This like why cancer occurs is like there will be normal cell, but if there's some genetic mutation which it encounter, uh, uh, generally they carry the genes from, uh, but due to the environmental factors, this will express the mutated version genes express, or the food we take, it cause the changes in the DNA, and then cause this cancer. That's a different subject from origin of life, because origin of life, because first thing is that we are discussing on from origin of life is that since we get a single cell from a precursor molecule, right, Nikhil? So that's
Yes, sir.
if you form the first cell, then later comes the cancer and all. See, there is no connection between these two.
Because I think like it should be somehow related, because like cancer cells also fight with the different means antibodies of our body to survive and then to spread itself, and like something like origin of life also, like cells survived different temperature and humid conditions which existed millions of years ago, and then that's how also life and microbes originated in that way, um, like as was given by that pool model, uh, food model, soup model in which life originated
Yeah,
after fighting so many harsh conditions of earth. So in that way, cancer cells also like somehow in body fights the different antibodies and the different supporting mechanisms. And then it originates, and so many variations also it shows in its existence in the body. Like our gland will have different gland, different type of cancer, same body may have. So when you relate that,
yeah. Actually, in cancer, why this is very harmful. Like they have mutation and it won't stay at one place. So they migrate to other place and cause the metastasis. This we call as cancer cell migration. So that's why cancer is so dreadful disease. Uh, and thing is that as you are thinking, like this is all hypothesis that previously you said that this cells originated in the primordial soup and all. This is all hypothesis. That's not true.
Primordial pool.
Yeah. So this is an hypothesis, but that's what, right? I from all this work from the scientists, I have shown that um, this there's no till now uh, cell formed from this precursor molecule from their even experimental regulated condition. Forget about prebiotic, unregulated. Regulated condition, they just made, Craig Venter made a bacterial cell with already existing cell, live cell only. They send the genome and then remove the previous genome. That's why it happened. But that's what this going on. Even when I'm in a school, like I feel like okay, this is how the life came from the many scientists. Even now they think because they're not given thought process for much of because they're busy with their works. Only the experts of origin of life scientists they know what the intricacies are.
Okay.
But we just think that okay, life came from this precursor molecules.
Thank you. Thank you,
Yes.
Dr.
And
Kotagiri.
like this is accepted fact that RNA is biotic in system kind of thing. It was accepted also that RNA was acting as a reagent in that prebiotic system for originating other life forms in its way. So RNA is related to means more specifically to the origin of life when compared to the other forms like DNA and other elemental particles like electron and then proton, those things also. So RNA, how is it like different and what structure it has that it is mainly accepted as the primary element for origin of other things?
Yeah. One thing is that as I
Yes,
said,
sir.
your question is done, Nikhil. Hello, Nikhil.
Yeah, sir.
My question is related to RNA. Like,
Yeah.
Yeah,
You are done with the
yes.
question.
Can you hear me?
yeah,
Yes,
is your question
my
completed?
question was related to RNA. It is acting as a reagent in
Yeah.
prebiotic system. That thing,
Yeah.
like how RNA is different from other elemental particles.
Yeah. I have um, already told that like you take suppose basically DNA is our genetic material. From there RNA after transcription we form we get the single strand of RNA. So forget about DNA now. So this RNA will give rise to with translation they give rise to protein molecule which fold and form different functions in a cell like whatever enzyme or it's a receptor or antibody, so many things. So uh, but for the DNA to unbind and perform its function it requires a protein. So initially scientists thought okay which one is there first, is it DNA or the protein? So which one form first? So it's like a chick and egg puzzle, like which one came first. So people thought, okay, this is not fitting to the hypothesis, so there won't be. But they looked for what is the other molecule. So they thought, okay, RNA is there, but this RNA until previously they have just thought might be the chance because from RNA it can form the DNA also, and from here the protein also comes. So it is in the intermediary stage. And with the finding of this Altman and Chet that these have the catalytic activity because protein have the catalysis activity and DNA have the genetic information. This RNA have both genetic information and the catalysis activity, but not to the full extent of every RNA will have this catalysis activity. It's not that way. So then they thought, okay, this is the why they have given importance for RNA is because of this reason. Both having the, so if you having actually genetic material, it
Yes,
can
sir.
transfer the information for the further generations. So it makes its copies. From that, it can make protein. And from, if you use an enzyme called reverse transcriptase, the RNA can form the DNA also. So which further, that's what the evolution theory that they say, that the RNA world theory says that now the RNA is there. Further, for future generation, it given, it made to the DNA. So DNA become the genetic material. So which we are having right now as a all. The information is stored in that. Yeah.
Yes, sir.
But
So
is it answer your question?
okay.
Why?
Yes, sir.
Yes. Thank you so much, Niket, for your question, and for Dr. Sashi for your reply, for your response. So it's basically a chicken or egg situation, and RNA is caught in between these two, in a sense.
Yeah.
So I would like to thank Dr. Shashi Kotagiri for his presentation and response to the questions. Can we all join to give him a round of applause?
I thank for the audience and then participants and then Bhaktivedanta Institute for giving me the opportunity, uh, particularly Vasudev Raoji and Rajpati for, and whole team who worked for Debshis Khan and so many, Dr. Debshis Khan. I thank each one of them, whoever supported for this to happen, and thanks for the opportunity.
Thank you, Dr. Sasi, for your presentation within the hectic schedule.
Thank you, Dr. Debshis Khan.